Hello and welcome back as we begin our look at the urinary system. Um, Over the next few videos, we will explore mainly the physiology of the urinary system. We will spend some time looking at the anatomy, but mainly the histology anatomy or the microscopic anatomy. In other words, how does the urinary system function? And so that is kind of where we're going to be spending the next few videos, is really looking at function and based on that function, how are we regulating homeostasis? So just as a review for us, this should be very obvious for you. If not, I'm concerned as we have spent some time in lab looking at the overall anatomy and structure of the urinary system. We know that the primary organ that is doing the majority of the work in the urinary system is this paired structure. Right? And these paired structures are what we define as being the kidneys. And I say doing the majority of the work within the urinary system because it is within the kidneys where we are filtering the blood and producing the filtrate. And so this is kind of an important focus for us on the urinary system. In fact, this is where we're going to be spending all of our time within the urinary system, is looking at the function within the kidneys. Besides the two kidneys, we also have a right and a left ureter. And the job of the right and left ureter is to transport the urine that has been produced within the kidneys down into our urinary bladder, for which there is one, and then the tube that is carrying that urine from the urinary bladder to the outside world is, of course, the urethra. And we've spent some time in lab looking at the histology of the urethra, as well as the urinary bladder and the ureters. And so, again, our focus for our look at the urinary system is really focused here on the kidneys. So, what is the function of the urinary system? And here we can see that we've got 1, 2, 3, 4. We have 5 main functions that are taking place within the urinary system. These functions are fundamental to what is happening within the urinary system. And almost all of these things, actually all of these, is taking place within the kidneys. And so what this has all in common is that all of these functions are happening inside of the kidney. And so all of these, all functions, occur within the kidneys. Right. And again, these functions is the filtering of the plasma within the within blood. And as we are doing that, we are eliminating waste. All right. We are eliminating by waste. We are referring to some nitrogenous waste, but we're also just referring to excess. Excess water, excess minerals, excess vitamins, excess molecules, anything that is in excess. Right? As we've talked about during the cardiovascular system, we know that the kidneys can also regulate blood volume. And so if you have too much water that is elevating blood pressure, the kidneys, through the filtering of the blood, can remove that excess water to lower the blood volume, which in turn would lower or aid in the lowering of blood pressure. We will find out that the main hormone that is involved in that is what we define as being renin. And we're going to talk about the renin, angiotensin, aldosterone system, what we call the RAS system. And so we're gonna be dealing with this in a lot more detail. So again, what is regulating that blood volume is that enzyme called renin. We also know from our look within the cardiovascular system that the kidneys secretes the hormone erythropoietin, and we know that erythropoietin is what stimulates pluripotent stem cells to go ahead and differentiate into red blood cells. And so red blood cell production is again regulated largely through erythropoietin being secreted from within the kidneys. And by the way, one of the reasons why that is because you do have chemoreceptors that are in the distal convoluted tubule, the macula densa. And one of the things that those chemoreceptors are monitoring as the blood is coming into the afferent and efferent arterioles is O2 levels. And so if it's noticing that the filtrate or the blood that is entering into the glomerulus is low in O2, then it will stimulate the production and the release of erythropoietin. And the last function that we have within the urinary system is the completion of the synthesizing of calcitrol. Calcitrol is a precursor, if you would, of calcitonin. And so here we have that tie-in not only into the cardiovascular system, but into the skeletal system as well. So, as I've mentioned previously, the primary function that is driving the urinary system is the removal of waste. And the majority of the waste that we are removing from the urinary system is nitrogenous waste or nitrogen-based waste, and that is typically in the form of urea. And that is a byproduct of the breakdown of amino acids. And so as proteins are being broken down and the amino acids are being broken down, we create this nitrogenous byproduct known as urea. And the urea is what really concentrates within the urine. We regulate how concentrated that urea is by adding or removing water. We do that because we are terrestrial. All right. But if we were a fish, we would actually secrete and release almost pure urea. We wouldn't have to convert it into urine. And the reason for that is because they're releasing that urea into the water, and the water then dilutes it. And so part of the process that we have within our urinary system is to go ahead and actually dilute that urea. All right. We also have uric acid. We have creatinine, which is the breakdown of creatine phosphate. We know that creatine phosphate is used in the replenishing of ATP. All right. So you have creatine kinase, which is the enzyme. This is going back to muscle physiology. Creatine kinase is the enzyme that removes the phosphate from creatine phosphate and adds it back onto ATP. Well, creatine phosphate is only good for so many cycles, and it wears down. And so when the liver breaks down creatine phosphate, creatinine is the byproduct, and we excrete that through the urinary system. So, when urea levels are elevated within blood plasma, that typically indicates that there is renal insufficiency, and by renal insufficiency, we are really referring to, depending on the stage, renal failure. In other words, the kidneys are not filtering the blood anymore. All right. Azotemia is what is clinically referred to as high levels of urea, and so you can see what the normal levels are for most people. One of the other things that we need to consider when we look at the urinary system is urethral or kidney stones. Right? So this urethral stone here that we're going to look at, it just simply is a kidney stone that has now made it down into the urethra. Right? And so if you look right here, you can see the kidney stone that is now actually down in the urethra. Right? So there is the kidney stone. Right? We refer to it here as a urethral stone only because it's in the urethra and it's no longer in the kidney. What is very different about this is if you look over here, right, so here's the kidney, here's a kidney. Right. What you're looking at right here is the ureter. Right. What you're looking at right here is the ureter. And that ureter is experiencing inflammation. Right. These are rather inflamed and swollen ureters. Right. And here is the bladder right there. And if you look right there, that right there is the kidney stone. Right, that right there is the kidney stone. Just some fun images for us to consider as we look at the urinary system. Here we have a normal kidney. Right there. Here we have a normal kidney right there. Here we have an inflamed kidney, and so this would be a kidney that would be experiencing kidney stones. This would be a kidney that would be experiencing kidney stones. We can see that even in this scan because if you look right here, you're not seeing the outline of the ureter, which means urine is not flowing through there. So that liquid creates density within the x-ray, and it doesn't allow the x-ray to pass through as easily. And so here you can actually see urine is flowing through, no urine here. So we know that this is the kidney here that has the kidney stone. Same thing here. You can see the ureter here, and then here you can actually see some more of that fuzziness that's graying it out. And that's because this is the guy here that has the inflammation or the kidney stone. This here is a malignant tumor within the kidney. All right, so again, if we look, what the heck? If we look right here, that there is the tumor. All right. One of the things that defines this as being a malignant, one of the reasons why we would assume that this is a malignant tumor, is because of the non-smooth edges. And so we can see that this here is an irregularly shaped, what we would define as being non-encapsulated tumor. And by the way, here you can see the normal kidney over here. All right, the other thing that you can see here is you can actually see the renal pyramids really well. In that scan. All right, but right there, that is the malignant tumor. So it's a non-encapsulated tumor that has a high degree of likelihood that it will spread. And usually when malignant tumors of the kidney spread, one of the areas that they spread to is the brain. And so you're looking at a lesion here and here. All right. If you look at that same spot, that lesion right there is this lesion here. That lesion there is this lesion here. And you can actually see some other areas of lesions as well. This is another area here. This is another area there. And this is not brain cancer. This is not brain cancer. When you biopsy this tissue is actually tissue that is resembling or almost nearly identical to what you would see in the kidney. And so this is not neural tissue that you see here. This is actually kidney tissue that has transplanted itself into the brain, and because it is cancerous, it is dividing uncontrollably, and it creates these lesions or these tumors within the brain. And as you can imagine, and you can actually see the distortion within the left hemisphere right here, where that left hemisphere is now being pushed against the right hemisphere. And so this would actually go ahead and then put compression pressure on this side of the brain, and it would have other implications as well. But this here is metastatic renal cancer of the brain. All right. This scan here is a benign tumor within the kidney. So you can see here we have very smooth edges. This here is actually encapsulated and contained within the kidney itself. And so this here has a low likelihood that it would actually spread beyond the kidney. All right. And so that was some little bit of a perspective as far as what the system looks like, both normal and diseased. On this slide here, we see the difference between the cortical nephron and the juxtamedullary nephron. This is something that I have tried to impress upon you in lab or through our lab activities. And so a cortical nephron is called a cortical nephron because the vast majority of the nephron is contained within the cortex. Right? There's only a small bit of the loop of Henle that will dip down into the medulla. Doesn't matter the nephron. All nephrons, that loop of Henle will at some point in time go down into the medulla. And the reason for that will become more apparent when we get into a little bit more of the homeostatic mechanisms of the renal system. Right. Over here you have a juxtamedullary nephron. The renal corpuscle sits lower. The proximal convoluted tubules and the distal convoluted tubules sit lower in the cortex, and notice that the vast majority of the loop of Henle extends down into the medulla. All right, hence the name juxtamedullary nephron. I'm also going to mention that this idea of a nephron is really composed of two parts: the renal corpuscle, which is where the actual filtering takes place, and then we have the renal tubules as well. And the renal tubules is where the reabsorption and the filtering, or the reabsorption and the secretion, takes place. And they happen in the renal tubules. Filtering is what happens in the renal corpuscle. And with that, we have the renal corpuscle. And so I actually want to kind of end our look of this first part of the urinary system with an understanding of what the renal corpuscle looks like. And we'll pick up with the renal tubules on the second video. And so this is the renal corpuscle. This is where all the filtering takes place. Right, as we have talked about and discussed, as we have looked at and discussed. That's not what I wanted. We know that we have a afferent arteriole that comes in, and that afferent arteriole is what is going to come in and then create or transition into the glomerulus, which is what you're seeing right here. So that glomerulus is a ball of capillaries, and then it then exits the renal corpuscle as the efferent arteriole. Right, so afferent in transitions into the glomerulus and exits the renal corpuscle as the efferent arteriole. This is one continuous vessel. This is one continuous vessel. And we'll get into a little bit more about why this is wider over here and this is thinner over here as we get into the regulation of this process. For right now, we're just understanding structure. And so the inside of the renal corpuscle, this is the glomerulus, this is the ball of capillaries. You will notice that it is surrounded, those capillaries are surrounded by specialized cells called the podocytes. Right? And those podocytes regulate filtration. And so they regulate the selective permeability of those blood vessels. In other words, if they are relaxed, they're going to allow for more permeability, more filtration. If those podocytes are more constricted, it's going to lessen the amount of permeability or reduce the amount of filtering that takes place within the glomerulus. The podocytes, because they line the capillaries, right, they form what we call the visceral layer of Bowman's capsule. And so these guys here form the visceral layer, directly attached to the capillaries. The outer edge of this structure that you're seeing right here, this is what we call the parietal layer. Right? Parietal layer because it is anchoring to the tissue surrounding the renal corpuscle within the cortex. All right? And so this is your parietal layer right here. The space between the podocytes, or this visceral layer, and the parietal layer, the space between that is what we define as being the capsular space. And it's within the capsular space, and here where filtrate collects. Right, now remember, we're defining this as filtrate. This is not urine that's coming directly out of the blood. This is filtrate. Right? This is water. It's ions, it is biomolecules, it is waste, it is other metabolites, drug breakdown, all that kind of good stuff. Right? All of that is now collecting within that capsular space. All right? Collectively, the parietal layer, the visceral layer, and the capsular space is what we define as being Bowman's capsule. The end of the renal corpuscle, where the afferent and the efferent arteriole are coming in and exiting, that is called the vascular pole. And then the end that the filtrate exits on is called the urinary pole. And it's the urinary pole that leads to the proximal convoluted tubule. Right, the proximal convoluted tubule. And so we're going to stop there for right now. When we come back, we're going to take a look at the renal tubules and their organization, and we will begin to progress into the homeostatic mechanism. In other words, how do we regulate this? How do we regulate what's happening within the renal tubules? And so that's kind of a preview where we are going. But for right now, we're going to stop there and give you some time to review this, to rewatch this, answer the questions, do what you need to do to make sure you understand the basics here. And with that, I'll catch you on the flip side.